an illustration of how the ribosome is. In the cytoplasm are special organelles called ribosomes that attach themselves to a molecule of messenger RNA, or the read, the sequence of RNA nucleotides, and using that information, beginning linking together amino acids to form a protein of molecule. So again, this is the ribosome, which has all the machinery to be able to recognize the messenger RNA and to translate that code into a protein or amino acids. Proteins are formed of amino acids. So this ribosome here has this A site that is able to recognize the nucleotides in the messenger RNA. So, but this site recognizes three nucleotides at the same time. And there are three nucleotides. The arrangement of these three nucleotides are the ones that codify for any specific amino acid. Here, an amino acid is represented for a ball, and here different balls with different colors means different amino acids. So once this A site recognizes these three nucleotides, it codifies or it decodifies this signal, and it knows what is the specific amino acid that requires these three nucleotides. Produce the nucleotides in the P side. These amino acids are going to be all linked together to form a polypeptide chain. That's what we see here. So what we see here is the mRNA enter to the A side, and in the P side the protein is going to be synthesized, and it's going to have a specific sequence depending of the mRNA that the ribosome is reading. Okay, so as we see here, the mRNA has a specific organization of the nucleotides and is recognized 3 by 3. 3 nucleotides are the ones that codify for one specific amino acid. And how are these nucleotides characterized? So this is this table that is called the RNA codon table, that where it tells you, depending of the nucleotide in the RNA, of the first position, second position, and third position, because recognize 3 nucleotides, what specific amino acid is going to be produced. So in the RNA, for example, if in the A site, as we saw in the previous image with the with the ribosome, if it's recognized 3 nucleotides, for example, U, then the amino acid that is going to be there is phenylalanine. And is if U, which is U, is going to be asparagine. So depending of the organization of those 3 nucleotides is the corresponding amino acid, and every protein has a different arrangement of amino acids. And it's very important here to recall again that the DNA, which is the original molecule, has only 4 nucleotides, which is not U, is T, thymidine, cytosine, adenine, and guanidine. The RNA has 4 as well, but instead of T, it has U, uracil, cytosine, adenine, and guanidine. And the proteins are made of 20 different amino acids. There are specific codons as well for stop, so the ribosome knows when to start and when to stop the protein synthesis. And these 20 amino acids are arranged in a very different manner for every specific protein. So, and this is what is called the process of translation, when the RNA that have this specific code is translated into amino acid. It recognizes 3 nucleotides in the RNA, and those 3 nucleotides in the RNA are codifying for a specific amino acid, and a chain of amino acid is what is called a polypeptide chain or a protein. It depends. If there is for example, a strain of around less than 50 amino acids, we call it a peptide or a polypeptide. But if it's longer than 50 amino acids, then we call it a protein. Now we see here, these are single amino acids, they form a polypeptide. There are different amino acids here linked together. And when we have more than 50 amino acids together, we call that a protein. And every protein, and as well some peptides, have a specific organization or three-dimensional organization. And this three-dimensional organization is going to help for the function of the protein. So the ribosome ensembles the very particular sequence of amino acids at the behest of a very particular sequence of RNA nucleotides, which were themselves encoded in the DNA that were given to us from our parents. So again, from the DNA to RNA, we have the process that is called transcription, and it's when the DNA goes to the RNA. And as well, we have from the RNA the process that is called translation, where the RNA is translated to have a protein, to be able to have a synthesis of protein. In molecular biology or in biochemistry, when we want to study different proteins or different DNAs, we use a tool that is called DNA cloning, and what we do is to manipulate or to generate a DNA that we can incorporate in bacteria to be able to study the characteristics of this DNA or the protein or different processes in the cell. So I put here a video. It's a very nice video that is going to explain to you how is the process of the DNA cloning, and what are the specific characteristics of the process, why it's used, and what are the procedure per se that is required for this.
DNA cloning is a method for isolating a particular sequence of DNA from a complex mixture of different DNA sequences. In order to clone a fragment of DNA, we need to insert it into a vector, usually a highly modified phage or plasmid that can replicate in a host cell. Generally, a plasmid vector contains three elements: a cloning site where the foreign DNA fragment can be inserted, a drug resistance gene which destroys antibiotics, in this case ampicillin, to allow selective growth of the host cell, and a replication origin to allow the plasmid to replicate in the host cell. To insert foreign DNA, we must first use a restriction enzyme to cleave the vector at the cloning site. Here, EcoR1 cleaves the palindromic sequence GAATTC to produce single-stranded ends called sticky ends. These can hybridize with any piece of DNA that has also been cut with EcoR1. Foreign DNA containing the sequence we wish to clone is digested with EcoR1 and then mixed with the cleaved vector. The sticky ends of the foreign and plasmid DNA molecules hybridize and then are sealed into phosphodiester linkages by the enzyme DNA ligase, creating a recombinant plasmid. Each of these recombinant plasmids contains the inserted DNA fragment, an ampicillin resistance gene, and a replication origin. A pool or library of circular recombinant plasmids is thus created. Each plasmid carries a unique fragment of foreign DNA, as indicated by the colored arrows. Next, the host cells, E. coli bacteria, are added to the recombinant plasmids. The cells have been treated with calcium chloride to make them permeable to DNA molecules. Through a process called transformation, a few cells take up a recombinant plasmid, while most other cells do not. The bacterial cells are poured onto a plate of nutrient agar containing the antibiotic ampicillin. With ampicillin in the agar, only cells resistant to the drug can grow. At 37 degrees, the cells will grow and multiply. Because they cannot move on the auger, each will produce a separate colony of cells. The non-transformed cells lack the plasmid with the ampicillin resistance gene and die. Cells that have a plasmid are resistant to ampicillin and survive. The replication origin allows the plasmid to replicate by using the host cell's enzymes. Plasmid replication is independent of host cell division, but plasmids are distributed to each daughter cell when the host cell divides. As the plasmids replicate and the host cells multiply, the number of copies of the recombinant plasmid is greatly amplified. The multiple daughter cells form a colony or clone. Because all the host cells in a colony are derived from a single cell, they all contain copies of the same recombinant plasmid with its fragment of foreign DNA. A variety of assay methods can now be used on the bacterial colonies to determine which contains the particular DNA sequence we wish to isolate.